I'm Laura Parfrey from the University of British Columbia, and today I'll talk to you about eukaryotes in the human gut, and I encourage you to think about what is the normal eukaryotic community in our gut microbiome. I'd like to thank you for inviting me to participate in this symposium remotely, and I hope to meet you all in the future. So we know at this point that microbes influence many aspects of host biology, including development, traits such as disease and body weight, hibernation. And it's not just mammals, it's not just humans, but microbes are ubiquitous on hosts. All large eukaryotes are colonized by microbes that really influence the fundamentals of our biology. But the human microbiome is not just bacteria. We know there are viruses, and of course eukaryotes that live in and on humans. Eukaryotes in the human gut have typically been studied as parasites, and of course there are many notorious pathogens such as Amoeba histolytica, which is the causative agent of amoebic dysentery and kills many thousands of people every year, as well as pathogens such as Giardia that are associated with diarrhea and can, in high loads, be associated with stunted growth and cognitive developmental delays. But not all eukaryotes are pathogenic. Some are commensal, and there's variation in pathogenicity according to the context of the human gut ecosystem, as well as context of other aspects of the intestinal environment. There are certainly many examples of eukaryotes that are beneficial. One of the best known in mammals is in ruminants and in termites. Eukaryotes help digest cellulose and so make a new aspect of diet available to these organisms. Host-associated eukaryotes are common across. Eukaryotes have been detected in association with soil animals. This is an image of a commentary I wrote for a paper by Geisen et al. In 2015, showing that there are many different eukaryotes associated with creatures in the soil. And so-called parasitic lineages make up 2 to 12% of all sequences detected in soils, freshwater, and surface oceans in recent papers. But they're not all pathogens, and I'd argue we should call them symbionts in recognition of the fact that we don't know how they influence the host in many cases. So how do we categorize symbionts? I'm going to go through an example that's quite far from the human gut to give us some context for thinking about symbionts and what we call them. So I live in British Columbia near the west coast. We can go out and find mussels in the intertidal zone. Mussels are colonized by endolithic cyanobacteria in many places. And there's a complex relationship between mussels and these symbiotic cyanobacteria. So usually the mussels are eaten by Physaster, one of the classic relationships in ecology, and the cyanobacteria bore into the shells and erode away the periostracum, the dark surface of the shell, leaving a gray, the gray shell. That weakens the cyanobacteria weaken the mussel shells and increase the mortality of mussels due to predation, so the shells are more easily crushed by the sea stars. So in this case, we can clearly call the cyanobacteria a parasite. They decrease the host fitness. But environmental context matters here and in many, many systems. If we look at present survival during heat waves, eroded mussels survive at a higher rate than non-eroded mussels. So the cyanobacteria parasite actually really increases survival of mussels following heat waves, which is a major source of mussel mortality. So in this case, those cyanobacterial symbionts would be considered mutualists. And just like the cyanobacteria and mussel system, many symbionts shift along that spectrum from parasite to commensal to mutualist, and it varies depending on environmental context. So the fitness consequences in terms of survival and reproduction are how we categorize symbionts into the mutualist parasite commensal spectrum, but as I'll emphasize again and again, that those labels must be applied in a more fluid manner than we typically do. So a given organism is rarely going to be one type of thing in all cases. And so myself and others have argued that we should consider the eukaryotes in our guts as gut symbionts rather than as strictly parasites and organisms that should be removed. So with that background, I want to go over two questions. What is the normal eukaryotic community in the human microbiome, and how do eukaryotes influence the gut ecosystem? I'm going to use bacteria to set up some context. We know that bacterial community composition in the gut has changed in industrialized populations. This is an example from Yatsunenko et al., looking at populations from Malawi, Venezuela, and the US. And here and in other graphs, I'll show you each point is the entire gut community of an individual, and the closer together 2 points are, the more similar their microbial communities are. So we can use this as a snapshot of how similar or how different the bacterial communities are. And we can see that Malawi and Venezuela have much more similar communities than either do to the US. And bacterial diversity is also lower in US populations compared to Malawi and Venezuela. And these patterns of reduced diversity are seen repeatedly in industrialized populations compared to non-industrialized populations. Why? Well, microbial exposure has been decreased by many factors of life in industrialized countries, including increased sanitation, dietary changes, eating more processed foods, as well as increased usage of antibiotic and other drugs. We know there are experimental results showing these effects, in particular, for example, for diet, where a so-called Western diet with high fat, high sugar, and low fiber reduces the diversity of the mouse gut microbiome in an experimental setting, compared to a normal mouse chow diet, which has plant material. And so these changes have consistently reduced the bacterial diversity in industrialized populations. That's led to what's called an epidemic of absence, or are missing microbes. So across studies from different parts of the world, we see that microbes that were previously really common, and these are examples from Malawi and Peru and Venezuela, we have microbes that were previously common across human populations that are much reduced in industrialized populations such as the US and Italy, shown here. And this has been termed our missing microbes, so microbes that are lost as a consequence of modern industrialized lifestyles. Well, what about the eukaryotes? Well, we know that, for example, the prevalence of helminths, while it's high in many parts of the world, has been greatly reduced in places such as the US and much of Europe, to the point where they're non-endemic anymore. And widespread campaigns to eradicate helminths and other so-called parasites are having an effect. So there we see population level declines in the prevalence of helminths in school-age children in sub-Saharan Africa here, as well as in other populations. And those are repeated in populations around the globe. So it's really imperative that we characterize the diversity of the eukaryotes in the human gut before we've completely altered those relationships, and before we can so we can characterize what is sort of the normal eukaryotic gut community. So here's a snapshot of the eukaryotes in the human gut from the population in Malawi that was taken from those same samples that I showed you from the bacterial community in work published in 2014. And we see that across this population in Malawi, we have lots of Blastocystis and Entamoeba are really common members of the gut, and a smattering of other organisms, including parabasalids, nematodes, and fungi. We tried to compare the eukaryotic community from the folks in Malawi to those in the US, but we really couldn't because almost nothing amplified. We had low levels of fungi and Entamoeba coli detected in 2 children. So the lack of amplification is indicative of a really low diversity of the eukaryotic community in the US. So I'm going to talk to you about another study, the Eukaryome of African children, that was spearheaded by Philippe Sansonetti and Pascal Van Esch of the Pasteur Institute, and really a broad collaboration of researchers around the world to investigate the communities, the microbiome of children living in the Central African Republic and Madagascar in the context of stunting. So this study recruited children aged 2 to 5 with no obvious signs of disease, so no acute diarrhea, no recent history of antibiotics within the last few months. And children were recruited from 3 groups based on their height Z-score. They were severely stunted, moderately stunted, or non-stunted, and so those correspond to malnourished or normally nourished children. And you can see a complete overview of the study in the study protocol published in BMC Pediatrics in 2018. So what do we find? What does the eukaryotic community look like in these children? We see many of the same players popping up as we did in that earlier, much smaller study, where you have lots of Blastocystis across both populations. Entamoeba levels are also pretty high, and a smattering of other diverse eukaryotes that we would typically see in parasitology textbooks, including Giardia, Hexamida, Interomonas, Ascaris, Trichuris, Pentatrichomonas hominis, for example, as well as ciliates that frequently colonize the gut. We see here and in many other studies that it's really important to interrogate results of sequencing-based studies to identify true residents of the gut, because we see a high proportion of our sequences come from dietary sources as well as environmental eukaryotes, such as flagellates and amoebae that are clearly coming in from environmental sources. But when we do that, we see these African children have a diverse community, and it's similar to what we would see in other parts of the world. Interestingly, these common eukaryotes, helminths and protists, are not associated with clinical variables. So this is you can see that the prevalence of Blastocystis, for example, is quite high, over 80%. Lots of Entamoeba, Hexamida, and yeasts and other fungi are common. In some of the eukaryotes, we see association by country, but we don't see associations with stunting, with markers of intestinal inflammation, for example, calprotectin, or with anemia. In these and other studies of eukaryotes from populations around the world, especially of healthy populations, we see that Entamoeba, Blastocystis, Indolimax, Hexamida, and helminths are commonly popping up. So it's giving us a picture of the eukaryotic diversity across the world. And we have limited data, but it appears that there's reduced diversity and prevalence of these organisms in industrialized populations. Blastocystis remains one of the most common, but its prevalence is reduced, and we see much lower diversity of other organisms. So what are the consequences of loss of diversity in the eukaryotic component of the human microbiome? Well, we're trying to answer that in part by better understanding how eukaryotes influence the gut ecosystem. And kind of the broader framework for understanding this question has been through the hygiene hypothesis and sort of the parallel to the missing microbes idea. We see around the world we have a rise in immune-mediated diseases as populations transition to cities and industrialized lifestyles. And we see from epidemiology and case studies that there's an inverse relationship between immune-mediated diseases and the prevalence of helminths worldwide. So echoing that missing microbes syndrome, if we're losing the prevalence of helminths and other eukaryotes, we see a rise of immune-mediated. And we're seeing increasingly in studies from different parts of the world that eukaryotic diversity is also lower in immune-mediated diseases. Here's an example for inflammatory bowel syndrome, where the carriage rate of common eukaryotes in the gut, Dientamoeba fragilis and Blastocystis, is lower in folks with irritable bowel syndrome compared to those that are healthy in Denmark. So healthy folks have greater rates, a more higher prevalence of so-called parasites. So it raises a question: can we improve our health by restoring our diversity? I've worked on this question experimentally with Katerina Yerku-Pomnikova out of the Czech Academy of Sciences. And she's developed model systems in rats to experimentally inoculate the helminth Hymenelepis diminuta, and as well as Blastocystis. So the question of this work is, how do eukaryotic symbionts impact the microbiome, and do those interactions directly or mediated through the bacterial microbiome influence dysbiosis or the development of disease? So now I want to tell you about a study that we did together with the Yerku-Pomnikova lab, analyzing what happens when you experimentally colonize hosts with Blastocystis by measuring the immune response and gut microbiome response. So we know that Blastocystis is associated with a change in gut microbiome from observational studies, and we typically see two patterns in healthy populations. One is a lower diversity in so-called control or Blastocystis negative folks compared to folks that are colonized with Blastocystis. And this is data from a study of healthy populations in Mexico led by Miriam Neves Ramirez, but it's been repeatedly seen in populations around the world. And we also see a difference in community composition, where Blastocystis positive individuals have a distinct bacterial microbiome composition compared to folks that don't have Blastocystis. And you can see that by the separation here in the blue and red dots. But we don't know if this is causal or not. So does Blastocystis cause higher bacterial diversity in a different community composition that are indicative of a healthy gut? Or in folks that have a healthy gut with higher bacterial diversity and a distinct composition, are they more likely to be colonized by Blastocystis because of differences in the gut environment might be more favorable to Blastocystis making a living there. So we tested this experimentally by inoculating rats with Blastocystis to see what happened. And so we asked, does Blastocystis increase the gut bacterial diversity and change the community composition in healthy rats? And we used an isolate of Blastocystis ST3 that was isolated from a healthy human with no gastrointestinal symptoms. We did this in 2 time frames and in the context of experimental intestinal inflammation. So we induced intestinal inflammation with DNBS to mimic Crohn's disease. And I'll walk you through the experimental design. And I want to reiterate, I'm saying we here, but this was done in the Jurku Pomjakova lab. So our really nice collaborators here. So we had our control group and our colonized group. We took fecal samples before Blastocystis colonization, after we colonized with Blastocystis, but before induction of colitis, and then after colitis was induced, to understand how does Blastocystis change the microbiome and the host immunology, host immune system and health parameters by itself, and then in the context of colitis. And like I said, we did this on 2 different time frames, a short-term experiments after, so Blastocystis was inoculated, and then 3 weeks later colitis was induced. In the long-term experiment, Blastocystis was inoculated, and then 3 months later, colitis was induced. What we found was that Blastocystis appears to be able to speed recovery from this experimental colitis after long-term colonization. And so we see signatures in weight change as a marker of overall health, where we see more severe weight loss right after the induction of colitis in Blastocystis colonized animals, but then a recovery on the 2nd day. And we were only able to monitor for a short time after colitis induction. Would be really exciting to follow up with longer term monitoring in future experiments. But we see the signatures of increased inflammation right after colitis, and then a rapid reduction indicating recovery as measured by several immune markers, including TNF alpha. So expression jumps up right after colitis induction, and then there's a significant decrease in the Blastocystis colonized group compared to the control 2 days after colitis induction. So again, indicating that Blastocystis can speed recovery after long-term colonization. We don't see an effect after short-term colonization, though, when there's no difference in weight change or in immune parameters. What about for the gut microbiome? So here I'm going to walk you through this setup. We're first looking at alpha diversity, or the numbers in relative abundance of different types of bacteria in the gut. And I'll remind you that in observational studies, we see that Blastocystis presence is associated with higher diversity of the gut microbiome compared to Blastocystis absence. And so here the Shannon index is a measure of the bacterial microbiome diversity with and without Blastocystis. And we see that before the Blastocystis is induced, so there's no difference between the rats at this point. There's no difference in the gut diversity, gut microbiome diversity. And we don't see any difference in gut microbiome diversity after Blastocystis inoculation either, so that we don't see any experimental change in the gut microbiome diversity when we experimentally inoculate Blastocystis, even after 3 months. We do see a change in only the long-term experiment and only a subset of samples from the colon after the induction of colitis, where the diversity is higher in Blastocystis colonize the individuals, then Blastocystis is negative, then this could indicate or be reflective of that faster recovery that we saw with Blastocystis in the weight and immune measures. But we see no change, like I said, in the gut diversity in response to Blastocystis. What about the community composition? So there was no difference before Blastocystis was introduced, but over time, starting at day 9 to day 11, we looked at slightly different days in the long-term and short-term experiments. We see those community composition differences developing and becoming more pronounced over time. After colitis is induced, we see those differences in community composition becoming even more pronounced. So really strong differences in community composition are observed in our model system when we inoculate Blastocystis, although we don't see differences in the gut microbiome diversity. And so what we see overall in this experiment is that introducing Blastocystis, which is a really common part of the eukaryotic microbiome in healthy individuals, may increase recovery from disturbance and is not associated with gut inflammation on its own. So that's consistent with what we see coming from studies around the world, where Blastocystis is more common in healthy individuals and is not associated with intestinal inflammation. And so with that, I'd like to thank collaborators, especially Philippe Sansonetti and Pascal Von Asch, who's now at the University of Lausanne, as well as Vincent Billy and Alison Mann from my lab and our collaboration with the Eukaryotic lab. And again, I'd like to thank you for inviting me to share this work. If you're interested in joining the lab or in chatting about any of these results, certainly feel free to contact me, and my email is below.